Hello everyone. tDNA is the segment of the Ti plasmid of the Agrobacterium tumefaciens which is integrated into the host plant cell. A number of genes and proteins are involved in the process. In this module, we will discuss the role of host plant proteins in the transfer and integration of the tDNA from the Agrobacterium cell to the plant cell. The learning objectives of this module are entry of tDNA into cytoplasm of plant cell, transport of tDNA into nucleus, integration of tDNA, regulation of expression of tDNA. tDNA complex composed of the T strand and Vir D2 covalently attached to its 5' end is transported to the host cell cytoplasm via the bacterial type 4 secretion system, which also transports into the host cell 4 other bacterial virulence effectors, Vir D5, Vir E2, Vir E3 and Vir F. The initial attachment of bacterium to plant cell is consolidated via the synthesis of bacterial exocellular glucans, leading to the biofilm formation. Some plant genes coding for glycoproteins and a cellulose synthase, when mutated, led to resistance to Agrobacterium transformation. These rat mutants suggest the significance of plant proteins in the initial attachment of bacterium to plant cell. Some more proteins present in the plant cell membranes are also required for this attachment, but their function is not known. In the cytoplasm, dynein-like protein DLC3 acts as a molecular motor for movement of the tDNA complex along the microtubular network. Besides the role of plant proteins for attachment and transport of Agrobacterium into the plant cell, several proteins have been identified that bind to tDNA and play a role in import of tDNA into the plant nucleus. This include Vir E2 and Vir E3 that are also transported to the plant cell via the T pilus. A plant protein Vir E2 interacting protein, that is VIP1, or the bacterial protein Vir E3 bind to Vir E2. Plant cyclophilin, which is a regulator of protein trafficking in plants, and a phosphatase PP2C that can dephosphorylate Vir D2 and inhibit its import to nucleus, are two regulatory proteins for tDNA import into nucleus. Vir D5 and Vir F are bacterial effector proteins that do not interact with tDNA but are transferred independently to the plant cytoplasm by the bacterium. Nuclear localization signals on Vir D2, Vir E3 and VIP1 are recognized by important alpha, which binds to them and carries the T complex to the nucleus. The plant protein VIP2 is a transcriptional activator and binds to VIP1 in order to get transported to the nucleus. Vir D5 and Vir F also have nuclear localization signals that transport them directly into the nucleus. Vir F is an F-box protein that brings about ubiquitination of VIP1 and targets it for proteasomal degradation. Vir D5 binds to Vir F and prevents the plant proteasomes from rapidly degrading Vir F, hence allowing the latter to degrade VIP1. The plant VIP1 protein is a transcription factor that is phosphorylated by the MAP kinase signaling pathway in response to recognition of pathogen by the plant. Phosphorylated VIP1 then enters the nucleus and induces expression of defense genes. Agrobacterium uses this VIP1 protein to transport tDNA into the nucleus by binding VIP1 to Vir E2 that codes tDNA. In addition, the Vir F effector protein degrades VIP1, thus preventing it from activating the plant defense response. Hence, Agrobacterium has evolved an ingenious way of suppressing host defense by putting VIP1 to its own use. Hence, plant proteins are involved in nuclear import. Besides VIP1, the plant proteins VIP2 and PBRP also play a role in Agrobacterium plant interactions. PBRP is a plant-specific transcriptional activator that binds to Vir-E3 in the nucleus and regulates genes involved in expression of the tumor phenotype in response to Agrobacterium infection. VIP2, which is brought into the nucleus by VIP2 Vir-E2 tDNA complex, is a transcriptional activator of histone genes and is required during chromatin formation involving the integrated tDNA. Integration of double-stranded tDNA into the plant genome occurs by using the DNA repair mechanisms of plants. In homologous recombination method, two host proteins RAT52 and single-stranded DNA binding protein and RAT51 involved in homologous DNA pairing and strand exchange reactions are required. In addition, an exonuclease MRE11 cuts the tDNA strands from 3' end to create 5' overhangs. RAT51 polymerizes on these overhangs. Similarly, in the plant genomic DNA, where double-stranded breaks occur, the homologous repair machinery is used to create 5' overhangs on which RAT51 polymerizes. RAT51 then mediates strand exchange by searching for microhomologies in the two DNA molecules. Finally, the tDNA ends are annealed to the plant genomic ends. In the non-homologous end joining or NHEJ pathway, Ku70, which is a double-stranded DNA binding protein that functions in heterodimer with Ku80, and a DNA-dependent protein kinase bind to the ends of double-stranded tDNA as well as the double-stranded breaks in plant genomic DNA. An X-ray repair cross-complementing protein 4, that is XRCC4, involved in bridging DNA for ligation and a DNA ligase LIG4 stabilize the NHEJ complex, bridge the DNA ends and ligate. An X-ray repair cross-complementing protein 4 XRCC4 involved in bridging DNA for ligation and a DNA ligase LIG4 stabilize the NHEJ complex, bridge the DNA ends and ligate the tDNA into the plant genomic DNA. Integration mainly occurs via the NHEJ method. The extent of tDNA expression is regulated by the launching of a defense response by plants. This defense is mediated by salicylic acid, a hormone known to induce expression of defense response gene. Gene silencing of tDNA genes is also brought about by the plant, either by methylation of tDNA or by the formation of small interfering RNAs that inhibit expression of the tDNA genes. For successful pathogenesis, Agrobacterium has to overcome these regulatory defense mechanisms. The module can hence be summarized as tDNA entry into plant cells is mediated by the type 4 secretion system of Agrobacterium. Several plant proteins are involved in attachment of the T pilus to plant cell. VIP1 is recruited by the tDNA and it binds to Vir-E2 that coats the tDNA. VIP2 interaction with the plant dynein proteins facilitates cytoplasmic movement of the T complex. Importance interact with nuclear localization signals present on Vir-E2 and VIP1 and transport tDNA into nucleus. VIP1 is activated by the plant defense signaling pathway in response to Agrobacterium integration with plant cell and is an inducer of plant defense genes. It is, however, hijacked by Agrobacterium to carry tDNA into nucleus. Second strand synthesis of tDNA takes place in the nucleus and it is then integrated into the plant genomic DNA by non-homologous end-joining DNA repair mechanism. Extent of tDNA expression in plant cells is regulated by siRNA mediated gene silencing or by SA mediated defense signaling. To summarize, we can say that transfer and integration of tDNA from the bacterial cell to the plant cell involves various proteins. A multitude of host proteins participate in the process. The process also involves the type IV secretion system and the DNA repair mechanisms for the integration of tDNA into the plant cells.